everybody.
This is the first time we've done a webinar. We really appreciate being our guinea pigs while we work through all of the kinks.
Yeah. So now that you can hear us and see us, I'm Erin McKenney.
And I'm Lauren Nichols.
We are both beginners at YouTube live streaming, and we're both researchers in the Dunn Lab at NC State University. You may also know of our lab as Your Wildlife. So today we want to give everyone a brief update on the Sourdough Project and answer some of your questions. We'll answer some of your questions as we go and save some of the others for the end.
And so for those of you who are new to Sourdough Project, so probably all of you know about it already, but it started out as an online survey that we distributed to the general public. We were hoping we would get a few sourdough bakers to answer some questions about their starters and maybe send us some. And it was incredible. Over 1,000 of you all over the world filled out the survey. You answered all kinds of questions about where your starters came from, how you maintain your starters, whether you have pets. It's really impressive what everyone was willing to answer for us. And not only that, 560 bakers then sent us actual live samples of your pet sourdough starters. So thank you if that was you.
And even considering the successes we've had with other citizen science projects in the past, like Cat Tracker and the Showerhead Project, the Sourdough Project participants, you are exceptional. You've submitted your questions, set up your own experiments, and even asked questions along the way that have helped us to develop and test new hypotheses. I personally think that this difference is probably because each of you personally takes care of or mothers your own starters every day, and you're invested enough to send us a piece of your world.
So if you're joining us, you probably already know that sourdough is awesome. But from a scientific perspective, sourdough is also unique and untapped study system. So even though humans have been baking bread for over 10,000 years, nobody really knows what the microbes in bread are actually doing, or even which microbes are there in the first place. So this is why it was really fantastic that we got such a diversity of starters sent to us.
So first, I'd like to share a map of where those 560 starters come from. So if you'll bear with me for just a second.
These slides might look familiar.
Yeah. All right.
Yeah.
So hopefully everybody can see that. Yep, there we go. So as you can see, 560 of you sent your starters from 17 countries around the world. Again, this is unprecedented sourdough diversity that's represented in this collection.
So unfortunately, as you can see, we didn't get samples from Africa or many other countries in Asia. So we do have some Western and Eurocentric bias in our sampling, and we want to acknowledge that. I think there's a lot of sourdough that we're missing and a lot of diversity we're missing here. But this gives us a great place to start. It's still the world's largest collection of sourdough samples. So if you did send us a sample, thank you, thank you, thank you. We could not have done this without you.
No, not at all. After we received your starters, we used DNA sequencing to identify all of the different bacteria and yeast in your starters. This process is kind of like taking the fingerprint of bacteria or reading the last names in a phone book to find out who lives in a community. And just check out the diversity that we found. Pretty much all of the commercial baking that we know of uses just a single species of yeast, Saccharomyces cerevisiae. But in your starters, we found over 70 different types of yeast. That's incredible.
So once we knew which microbes were in each of your starters, we could start to compare the communities across starters, and using the information that you had sent out, it had answered in all your questionnaires. So where your starter lives and where it was created and how it's maintained, and to try and figure out why we see certain microbes in some starters and not in others.
So far, we think that yeast tend to vary depending on your geographic location. We think this is because of the climate where you live. We found in other studies that geography tends to be a pretty good predictor of fungal diversity. Yeast are a type of fungus, so this makes sense.
And the bacteria seem to have a different story. They don't seem to follow the same geographic rules that the yeast do. And this is true also with other studies we've done with bacteria, where we found that the fungi follow geographic patterns and the bacteria aren't explained that way. Instead, the bacteria seem to vary depending on factors within your home, like what kind of flour you feed your starter, whether you keep your starter on the counter or in the fridge, and even whether the baker reported to be a male or a female. So we're still tweaking our models. This is all really preliminary, but it's starting to give us a picture of why we see certain yeast places and why we see certain bacteria in other places.
Yeah. The bacteria are also incredibly diverse, although most produce lactic acid or acetic acid. So acetic acid is the main ingredient in vinegar, while lactic acid is more often found in buttermilk, yogurt, and other fermented dairy products. Lactic acid tends to taste less sour than acetic acid and even a little buttery, some people say. So acid production, no matter what type of acid, is really important in sourdough for two reasons. First, it gives sourdough its unique tangy flavor, but even more importantly, acid prevents the growth of microbes that might make us sick. So this also means that bacteria that produce the acid actually help to extend the shelf life of bread. In the Sourdough Project collection of starters, your starters, we identified 3 different types of sourdoughs based on the types of bacteria your starters contained. Some were completely dominated by lactic acid bacteria. Some contained lactic acid bacteria and some acetic acid bacteria too. And some also contained kind of a mystery type of bacteria that we were unable to identify.
So let's see, move the slide forward. So here we can look at 2 samples a little more closely. So these are 2 different samples submitted to the project, and you can see the microbes that we found in each of them. So over here on the left, you can see starter 867. And this is what we may call a typical starter. So its dominant bacteria are microbes are a combination. We often think of the microbes in sourdough starter. You can see that in terms of yeast, it's dominated by Saccharomyces cerevisiae. And for the bacteria, it's dominated by Lactobacillus sanfrancensis. And these are the 2 most typical microbes that had been identified in starters before.
Yeah, prior to this project.
Yeah.
So in contrast, when you look at the right, starter number 862 contains mostly a different type of yeast known as Kazakhstania unispora and 2 totally different types of lactobacillus, Parliamentarius and brevis. This is interesting not only because these microbes hadn't ever been identified in starters before, but also because these 2 different starters currently live less than 30 miles from each other in North Carolina.
So as a side note, we actually got both of these participants to give us a new sample of their starter, and we recently baked bread with both of them. So if you are ever in North Carolina, check to see what we're up to, because we might be doing an experiment like that. Yeah. So this figure is also exciting to show you because it brings us to 2 really important updates we haven't shared anywhere else yet. So as you may have noticed, we list 2 different strains of Saccharomyces cerevisiae, so strain 1 and strain 2. And a lot of you were curious about what this meant. And sent us questions, and we were really curious too. So we went out, we wanted to find out whether these were really 2 different types of yeast or whether they were just the same yeast. So we had to turn back to DNA sequencing. You might remember when Erin was talking about how we did our sequencing, that we were only reading short pieces of DNA from microbes from all the samples. So this gives us a sort of brief overview of who exists in the community, and kind of like having just the last name of all of the different microbes. So this time we went back and we isolated each of these specific strains, so Saccharomyces cerevisiae strain 1 and strain 2, and we looked at longer sequences of DNA. To use the same analogy we were using before, if the first pass of DNA sequencing gives us the last names, this time we got their full names. And as it turns out, what we originally thought were 2 different strains of Saccharomyces cerevisiae is all really just the same strain. So if you have strain 1 or strain 2, it's all the same thing.
Update 2. Many of you are also curious which Lactobacillus strain corresponds to Lactobacillus sanfranciscensis. We used the exact same DNA sequencing technique that Lauren just described to get the full name of the bacteria to figure out which of the Lactobacillus sequences really belong to the species sanfranciscensis. And as it turns out, what we originally listed as Lactobacillus species 7 is actually Lactobacillus sanfranciscensis. So now you can all revisit your samples on the map, see if you have Lactobacillus species 7 listed, that means you have Lactobacillus sanfranciscensis in your starter. So now we can actually answer, finally, everyone's burning question. Is San Francisco sourdough bread really that special? So drum roll, please.
I get to answer this one. Nope. Sorry, San Francisco. So remember the global data set. It turns out the Lactobacillus sanfrancensis is not geographically bound to San Francisco at all. We found it everywhere. So this is a map of all the samples that had it in it. So San Francisco bread isn't quite so special after all. At least it isn't special because of this specific bacteria. It still could be special for some other reason, I guess.
But that's okay, right? It's not really about who's in the starter, but more about what they're doing for your bread, right?
Sure. I suppose.
So through all of this, we're learning a lot about what microbes live in starters, but as bakers, we also want to know how those microbes are influencing the smell and taste of bread. To try to figure this out, our partners at Tufts University in Boston grew live cultures of yeast and bacteria from each starter so that we could study what each microbe needs to live and how they interact with each other. They even have started to pit different microbes against each other to see who wins when, if multiple types of microbes kind of are introduced to sourdough starters at the same time. These studies are helping to understand how, out of all the microbes in your home, your sourdough grew its own specific community. For example, we've identified 2 different microbes that compete for a specific type of sugar called maltose that I'm now learning a lot of bakers add to their bread doughs.
So instead of a trophy, they're getting maltose.
Mm-hmm.
Cool. So having live cultures also lets us measure aromatic compounds. So this is what we perceive as smells and aromas. They do this with a machine that they call a nose, which I still haven't seen in person yet, but I'm really looking forward to going up and visiting so that I can see. And this will let us figure out how the microbes are making our bread smell and taste delicious. So this is research that's happening right now.
So more updates to come. But for now, that's all the updates that we have. So it's time to answer some of your questions.
Yeah, so a lot of you sent in your questions ahead of time by email. So we'll start there. Let's see, what are some of the questions? So throughout the project, we've gotten probably the most frequently asked question, unfortunately, is I sent you a sample, but I don't see my starter profile on the results map. What happened to it? If you shipped us a sample and your sourdough and it's not on the map, I'm really sorry. A few things might have happened. We might just not have gotten your package. We got a few packages after our deadline, and because of the nature of the sequencing we're doing, we have to put all of the samples on the sequencer at the exact same time. Otherwise, you can't compare them. So if we got your sample even a day afterwards, we couldn't go back and sequence it again. So I'm really sorry to the few of you who sent in your packages and they just got to us a day or 2 late. There were a few samples that came to us in really bad condition. Their labels were mangled. We couldn't match to your information. They had no email address, so we couldn't link it up to the data in the questionnaire. And then I think I've emailed each of you specifically if we had a specific sequencing problem. So there are a couple that went wrong. Sometimes things just don't work, but I think you would have gotten an email specifically for me saying that that's what happened.
Yeah. So now we have a question from Bob Parsons who asks, does the flour you use have an important or even the most important effect on sourdough microbes? So this is another one of those really popular questions that we've been asked, aside from like, I don't see my sample, right? There are a couple like most popular what greatest hits questions we get over and over again. So flour, Bob, thank you for being a voice piece for the masses, because a lot of people are interested in this question. It turns out that what we do know is if you feed your starter bleached or unbleached white flour, then your starter will contain a greater variety of microbes than if you fed it whole grain flour. Now, I think this is because white flour contains a lot more starch than whole grain flours, and starch is really easily digestible by lots of different microbes. It's such an easily accessible nutrient. So it's, anyone can use it. But we haven't formally tested this yet.
Yeah. So let's see. Kim Starr from Hawaii sent us an email. I'm going to paraphrase a little because it's long. So Kim pointed out that Saccharomyces cerevisiae seems to be fairly common in starters and wondered whether this is due to contamination from commercial yeasts. So we got this one a couple times as well. People who found it in their starter and were like, but I started mine from the wild and it still has it in it. So while Saccharomyces cerevisiae is a type of yeast sold commercially to bake bread, the species is also found naturally as a wild yeast. So it's still possible to start a wild culture that includes these. It doesn't make it any less wild.
Or spontaneous.
Spontaneous. And there's also evidence that once Saccharomyces cerevisiae does colonize a starter, it tends to dominate, become the dominant yeast. So it's possible that the microbial communities in a starter will shift after Saccharomyces cerevisiae is introduced, whether that introduction was intentional or unintentional.
Maybe it's just a heavyweight champion in all those, prize fighting competitions that we're setting up.
So it seems like Kim had a couple other questions. So the second question is, when you list species such as Lactobacillus species 2 or Lactobacillus species 3, is it because the species is not named yet, or is that you can't tell to species level? So for any given strain, this scenario may be true. Either of those scenarios may be true. For example, in our update about Lactobacillus species 7, when we went back and did more sequencing, we realized that it was actually species San Franciscensis. So as we continue to do culturing and further sequencing on some of these strains, some of the species level classifications might be resolved, and we'll try and update those and as we can.
Mm-hmm.
And Kim's final question is, does periodic refrigeration cause Saccharomyces cerevisiae to thrive over other species of yeasts in starters? We don't know yet. That's also going to be a common response, unfortunately. So the beauty of doing a broad survey of sourdoughs is that we get such a great diversity of samples. The problem with it, though, is that it makes it really hard to ask really pointed questions like yours. So far, the information we had about whether each sample was refrigerated has not explained the yeast in the starters. It does look like it might make a difference to the bacteria, whether you refrigerate it or not, but we aren't really sure yet how the temperature affects the species in your starter. So we have some hints. Hopefully we'll be able to resolve some of it, but most likely we won't have a very concrete answer with this experiment. We'd probably have to do a more pointed experiment or ask a different set of questions.
So once again, your questions are helping to fuel future research, right? Our next question was sent in by Matt Sullenberger. Matt says, I have a couple of questions. First, is it possible to download a full list of the currently identified organisms from my sample? I see them listed on the map, but it would be easier to have it in a file, and I wasn't sure if that was the complete list or just what dominates. Second, I'm wondering what you know about sourdough and celiac. My wife was diagnosed with it today, and bread is her favorite food. Matt then goes on to mention a paper published in 2007 that showed that certain strains of bacteria can reduce the gluten content of sourdough below a certain threshold, and he wonders how skeptical he should be of these results and whether having these species in his starter might reduce the gluten level in his bread. So backing up to answer your first question, we don't currently have a full downloadable list that we can share, but we will definitely keep you posted if and when these data become available. However, it is possible that there were other microbes that we identified in the sample that we didn't report. For fungi, we only reported yeasts, and for bacteria, we only reported those species that belong to the class Lactobacillus, as these types were the most likely to contribute to sourdough bread rise and flavor. In most cases, these did encompass the majority of what we found anyway. With regards to the celiac question, we've heard many versions of this question as more and more people follow gluten-free diets. First, it's important to point out that that paper you mentioned from 2007 not only tested specific species, but specific strains within each species in their lab experiment. So even if you have the same species in your starter, there's no guarantee that they're performing exactly the same digestive functions, much less to the same extent that they were in that experiment. I'd be willing to bet that the microbes in sourdough starters are able to digest gluten to some extent, but I wouldn't feel personally comfortable recommending bread with any level of gluten to someone with celiacs. But that said, it's pretty incredible how much we're learning about microbes in general these days, and especially the microbes that help digest our food even before we consume it. So I don't know, who knows? Maybe the diversity of microbes in sourdough starters will help to make bread safer for everybody to eat someday.
I hope so.
Yeah, so good luck with that. I know celiacs is no joke.
Yeah. So let's see, next question from David Winston. Will you please retest a subset of submitted starters to determine if microbial makeup remains stable over time? Oh my goodness, we would love to do that. We're both really interested in the succession process and how starters change over time. Obviously, this experiment wasn't to test that specifically, but if we find the time and the resources, that would probably be our chosen next step. So
yeah, if any of you have some money, want to toss it our way, or let us know about some funding resources,
and if we find other resources, then we will definitely hit you up for your samples so that we can test them in this way.
Yeah, absolutely.
So stay tuned. We'll work on it. So Doc Doherty. So Doc Doherty sent us several questions about how the feeding regime for a starter alters the microbes it contains and how, as well as its microbial growth rates. So unfortunately, our questionnaire didn't include detailed questions about ratio of starter to water to flour or the length of time that starters were refrigerated before being refreshed. For this initial study, we were really focused solely on identifying the microbes in starters and comparing the starters from across the world. But we would love to follow up if we're able to get more resources and have more time. Some other place. I mean, there's so much to learn about sourdough and microbes in general.
It's been incredible how finding answers to the initial questions has really just helped develop more and more questions that we want to test. It's a project that could go on for decades, really. So next we have a question from Phil, who asks, why does my sourdough get more sour the longer it incubates? I just made a loaf after the starter had sat on the counter with a towel over the bowl for 2 whole days, maybe 3. The starter smelled really sour, more sour than usual.
So I saved this question to give to Erin because this might be her favorite question in the world to answer. So thank you for asking this. It's like we set her up for it.
Yeah, microbial ecologist in the house. So yes. You might remember that we pointed out that all of the samples had bacteria that either produced lactic or acetic acid, right? So the longer that you let your starter go unrefreshed, the more time the microbes in your starter have to eat the resources you've given them, like the starches and the flour. And then the denser that those microbial communities will become, right? Because your microbes are dividing, filling up that starter. A longer incubation time also gives the bacteria more time to digest starches and flour to produce acid. So we actually have a really fun graphic that we developed for this project.
So I don't know how feasible it is and whether this is, let's see if this is looking forward or backwards for y'all. I'm going to assume it's right.
Yep.
Right side around.
Yep, we're good. So you can see when you make a starter, you have bacteria and yeast that are coming in from the environment and other sources, as you now know, right, to digest the starches in the flour. And those bacteria are producing acid while the yeasts are producing the aromas and the carbon dioxide that actually give the bread its flavor and rise, right? But those acids are what are giving the bread its really tangy flavor. That's why we call sourdough sour, right? So not only are those bacteria going to produce more acid the longer that they have to digest, but also the temperature that you're incubating and proofing your loaf is going to change kind of which microbes are more active. So if you've ever made a bread that's proofed overnight or for a long period of time in the fridge, that tends to really give you a more mild and complex flavored loaf because it's favoring the yeast fermentation and the production of those aromas. But fill your loaf of bread on the counter. Those warm temperatures really favor the activity of bacteria in the starter, and that means you're getting a lot of acid production. So as you saw, the longer and the warmer that your dough is actually proofing, the more acidity you're going to taste in that bread. Love, love, love that question. Keep them coming.
Let's see. Winston asks, what microbes are typically found on flour mites? So I had no idea when I saw this question, so I actually had to do a bit of research to answer this one. And I did find some papers about it. What we found was it turns out that flour mites have several types of bacteria, both in their guts and in the reproductive tracts. But we really have no idea how they function, how they might be contributing to the microbial composition of flour, how they contribute to the microbes that end up in your sourdough. So if anyone is a scientist out there and feels like studying microbiome of flour mites, come talk to us.
Yeah, where entomology meets baking, right? We have the flour, the bugs in the flour, the bacteria in the bugs in the flour. I mean, it's fractal.
Yeah.
Our next question is from Jeff Gossett in California. Jeff writes, are there any pathogenic bacteria in the starters? Is smell a good indicator of the health of the starter? And are there any residual probiotic bacteria left in the bread after it's baked? So I'll go through those three questions. In answer to the first one important thing I'd like to point out is that most bacteria, just across history, have been studied in the context of medicine. So there's a definite bias in the information you're going to get about any type of bacteria if you Google it, right? If you just type in any bacterial species randomly, you have a pretty good chance of running across the word pathogen in your search results. But that doesn't mean that most bacteria cause disease, especially in a sourdough starter, right? This is very context dependent. And especially considering that you're going to bake your bread before you eat it, right? So the next question, whether smell is a good indicator of the health of the starter. First, in this project, we weren't actually able to get into questions of which microbes are going to make a starter happier or healthier than the others, right? Because again, this first project was kind of a census of who's in each starter and not necessarily what they're doing or, signs of health or not. But at least in my kitchen and my experience with starters, a happy or healthy starter will typically bubble and kind of double in size within 8 to 12 hours after it's fed. And that time may vary depending on where you live and, the feeding habits, as you know. But we're still learning which aromas indicate healthy starters or which microbes live in your starter. However, again, in my experience, a starter that smells like alcohol or even acetone is definitely a hungry, sometimes a pretty hangry starter, like ready to be fed if you smell alcohol or acetone. So you can use it to kind of diagnose like, oh, this poor little starter needs some flour stat, right? And for that final question about whether there are any residual probiotic bacteria left in the bread, nope. They're dead after it's baked. Nothing survives the oven. But they don't really need to if you think about it, because they've already made the bread rise and digested the flour to create vitamins, the acid, and those other nutritional benefits. So you use them, then you lose them.
So I think I do need to back up 1 second. There's potential that some microbes survive the oven. There are extremophiles in your home. I feel like I can't be in this lab and not point that out.
Anything's possible.
Probably not the ones that were in your bread. They're probably adapted to the starter and not to the oven.
Thank goodness.
So let's see, another question. Teresa in New Hampshire sent us an email. She shares a headline about Lactobacillus Plantarum, which claimed that this probiotic can help to improve the skin of adults with acne. Teresa's starter contains Lactobacillus Plantarum, and she's wondering if she could use the starter as a topical treatment for skin conditions such as acne or athlete's foot. So we're not doctors, first of all, so we can't really prescribe anything. One important point, though, about the headline is that the acne study was likely completed using a pure culture of Lactobacillus Plantarum. That's mostly the way that these sort of trials are performed. And while your starter might contain this species, your starter probably also contains other bacteria and fungi as well. So while specific probiotics may be beneficial by themselves in really specific lab-controlled settings, I can't recommend using the sourdough as any sort of treatment for any kind of condition.
Yeah, definitely an interesting thought there.
Yeah, definitely interesting.
And finally, we have a shout out for Sadie Sheffer, who wrote a while ago, I run a gluten-free sourdough bakery in the Bay Area, and I've got a dilemma. We're trying to relocate our operation, and I'm worried that the taste of our bread will change if we move too far. We just relocated from San Francisco to Berkeley with success, but I'm wondering if I can safely leave the Bay Area. Is this something you might be able to help me with? My plan right now is to send starters to a few people in a few different locations and have them feed each starter daily for about a month, and then send us a loaf of bread baked with those starters at the end that we can taste. This is awesome. This is exactly what we were talking about of, the unique investment of the Sourdough Project participants, because Sadie actually went on to run this experiment, and she found that one of those loaves of bread changed in smell and the other didn't. So well done, Sadie. And as long as we're highlighting Sadie's email, maybe you want to connect with Matt's wife who has celiac.
Yeah.
It's like a gluten-free bakery.
She can still enjoy her bread. But yeah, this is the kind of experiment that would probably be like another next step for us. So a lot of you had questions about what happens with your starter if you move
or travel with
it, or send it to someone, a sample of it to someone else, and they live somewhere else and treat it somewhat differently. So that would be a more experimental approach,
like a transplant experiment. Yeah. So again, hopefully coming soon if we can find the resources.
So I think that's the end of our emailed questions. It seems like we've gotten a few questions submitted online. So bear with us while we sort through those for a second. Let's see. How much will a starter change over time? I sent mine into the project in January 2017. I wonder if the species listed would be different if I sent a sample in today.
That's another one of those that we would really, really love to be able to actually test experimentally. Yeah, because I could imagine, again, if you moved, if you start, if you're changing your baking habits or where you keep your starter, a lot of those, if you start feeding your starter a different type of flour, we do have evidence from this global data set that a lot of those factors do significantly impact what microbes are found in starters, but not across the life of a single starter. So those longitudinal experiments are things that Lauren and I would really love to dive into. Yeah,
right now we just have a slice in time with each of your starters. So even depending on how hungry it is and whether the microbes are outcompeting other microbes at that point, it's probably the same composition, but maybe the percentages could be different.
Yeah.
Or if you've made drastic changes since inception, there might be composition changes as well. Given a couple or more starters in the same refrigerator for many months or years, is there a risk of one taking over the other? Would the two starters tend to remain unique?
Ooh. Okay, so anecdotally, again, we haven't tested this. Anecdotally, I think partially that would depend how tightly you screw the lids onto your jars, or seal the containers if you keep your starters in Ziploc bags or whatever containers you keep them into. So from experience, at some point I had 14 different starters in my fridge. Now I'm down to 3, so I'm doing a little bit better. Maybe I have 6 since the gala. I've had a number of starters and a lot of lacto-fermented produce in my fridge all at the same time, and kombucha. And the only instance of kind of cross-contamination that I've ever run across was not between starters. And again, this is anecdotal because the breads that I baked from those starters, they continue to taste distinct. So I'm kind of guessing from the end product that they're still unique microbial starters, right? But I did have a really interesting a jar of lacto-fermented pickles that I opened one time, and the mouth of the jar was plugged with this like really slimy, thick plug that was just like a kombucha mother. So I think at some point I might have had some kombucha SCOBY crossover into a jar of pickles. We didn't eat that jar, even though it looked just like a kombucha plug. But suffice it to say, I don't think that I've ever had cross-contamination between starters.
Yeah. Again, we don't have like actual sequence data to back this up.
Just anecdotes from the kitchen.
Ross K says, I successfully used my starter to carbonate a beverage instead of beer brewing yeast I usually used. Are there differences in how much alcohol a sourdough starter produces relative to a brewing yeast?
Ooh.
So Saccharomyces cerevisiae is the yeast that most people use. I mean, it is brewer's yeast. So depending on which yeast you have in your sourdough starter, it might be a Saccharomyces cerevisiae. My guess is that it really is based on more of the environmental, like what you're giving it, how much and how long you're letting it ferment, how much alcohol it's going to produce.
Yeah, if you give it a liquid wort, right, made with a lot more soluble starches and sugars like they would have in a beer making environment, then maybe the same yeast could start to produce alcohol instead of those aromas.
Mm-hmm. Yeah. But for a lot of these other yeasts, they haven't been characterized yet, so we don't know.
No.
So if you have something in your sourdough starter that's different from Saccharomyces cerevisiae, you know better than us.
Yeah, maybe you'll have the next really interesting bread beer instead of beer bread.
And we had the part in the questions where everyone couldn't hear us. That was sad.
But we fixed it.
Yeah. So I'm just going to read all the questions. If it's something we've already answered, we can just move on. Is the health of a starter affected by whether it is fed 100% whole grain versus refined flour? So you answered that a little bit before.
Yeah, I think there are two different concepts there. One is, well, the main concept there is health, right? What defines a healthy starter? And I don't know that we can answer that with our current data, right? To me, the health of a starter is more based on like how does it act in your kitchen and what kind of bread does it bake, right? If you're happy with it, then you might call your starter healthy no matter what microbes are in it. We do know, however, that the diversity of microbes that live in that starter do vary depending on if you feed the starter with a white or a whole wheat flour. So it does seem like white flours, bleached or unbleached, tend to kind of fuel a greater diversity of microbes than do whole wheat or rye, like those whole grain flours.
I think we answered that question already. So I'm just going to skip that one. Well, how does temperature affect the starter? If I keep it in the fridge mostly, is that creating a different community? So mostly for the yeast, that doesn't seem true, but it does seem true for the bacteria, and we're still trying to figure out in what ways.
Yeah,
is the short answer.
And again, we could get at that over time with a longitudinal study. We could speculate that maybe you would have more changes in a starter kept open on a counter than closed in a fridge, right? In addition to those temperature differences, but we have no idea currently.
Karen Hart asks, do you know what temperature to incubate the dough to make a good sour flavor?
So this is coming back to Phil's question that I really enjoyed answering. So if you want it more sour, I would suggest that you proof your dough for a longer period on the counter or in a warmer environment, because that really fuels a lot of bacterial growth.
But it also might depend on which cultures you have. So even when we had I mentioned that we did an experiment where we actually did another experiment where we had 3 different starters, and they had all different microbes in them. And we had bakers bake the bread the exact same way for all of them. And some were more sour than others, and they'd been maintained the same way. So some of it has to do with what you do, and some of it just has to do with who you have.
Yeah.
My understanding is that Saccharomyces cerevisiae will ferment relatively simple sugars, but not more complex carbs like starch.
Is there another organism in starters that breaks longer chain starches into simpler sugars that Saccharomyces cerevisiae can ferment? Let's see. So I know that in gut microbial communities, often there are different types of microbes that can kind of attack and digest different size and complexity of compounds. So certainly there are microbes that are able to snip apart more complex fibers, right, or starches into sugar units that then can be used by other microbes like Saccharomyces cerevisiae. But I haven't personally studied those kind of food webs or interactions in sourdough starters. It wouldn't surprise me. We just don't know who's there or even what all these different types of microorganisms are doing in those starters yet.
Oops, I just accidentally. Do you have anything to say about the nutritional benefit of eating the starter itself, like eating a spoonful?
My concern there would be if you had any microorganisms in there that could like cause an upset stomach or something like that. If they can live on sugar in the outside world, you may not want to put an entire spoonful of a diversity of microorganisms in unbaked bread, kind of like eating cookie dough, right? So I might not recommend that. Especially a lot of the benefits of that microbial fermentation have to do with the acid production, but also vitamin B. And vitamin B, I don't think that it's really broken down at the temperature that the bread is baked. So I don't know that you're getting too much more benefit eating a spoonful of raw starter. If you want to eat live microbes, I might recommend a yogurt or lacto-fermented vegetables instead. Yeah.
It's a little less diverse, and you can have a better. It's been tested for many thousands of years, being eaten in that form, and so we can comfortably recommend that.
Yeah.
So Scott Clausen, I respectfully disagree. My bread continues to develop flavors for a couple days after baking, mostly tang associated with the bacteria. There are enzymes that, when combined with flour and water in a relatively neutral pH, convert starches to digestible sugars, I think.
That sounds reasonable to me.
And yeah, breads do change. I mean, foods in general will change and develop flavors as they settle. And so also your bread, after you've baked it, will be colonized by microbes. So it's not a sterile, and as soon as it comes out of the oven, the second it hits the air, it's being colonized. So it's true. There are new microbes colonizing and digesting those starches. So it will continue to change flavor. And it's possible that there's still some fermentation happening at that point.
Yeah, especially if you're an active baker or fermenter, so then your home might be enriched with those fermenting microbes. Yeah.
Yeah. Austin Lee asks, can you comment on what extent a colony locks in the distribution of species versus adapts to changes? How difficult is it to completely change the dominant species just by environment changes? So again, this is one of those where we just have a snapshot in time from lots of different starters, and we have no idea what they look like a day before or what they look like a day after.
The only inferences we could possibly make about membership is based on the activity of starters, right? So at the point that you have a starter that you want to bake with, it is fairly stable, right? You're not baking with your starter like 2 days after you start mixing flour and water. So there's a stability in the activity of the microbes, but we don't know yet if that reflects a stability in the membership of that community.
And the only other sort of hint we have is that it seems like, so the yeast we saw in the samples does seem to be determined to some extent by where they are globally. And a lot of this, we heard a lot of stories about, well, my starter was originally from here, but now we're here. So if starters were remaining sort of true to what they were initially, it would be really hard to see that global geographic distribution. So again, this is just sort of hypothesizing, but it seems like there might be some changes happening if you move geographically. But again, we'd have to go back to an experiment more like Sadie Schaefer, where we send a single starter to lots of different places and have people treat them the same or differently to do it more experimentally to know an answer to that question. Off topic here, but I'm making vinegar from kombucha, basically letting it brew until it's very dry, especially for pickling vegetables like cucumbers.
Ooh. So keep an eye out. You might get one of those plugs, Malcolm.
Also, one of our collaborators who's doing the culturing work at Tufts right now, her other baby project, and by baby I don't mean a small project, but her baby, is studying kombucha. So you might hear about that one soon if you're interested in kombucha and brewing kombucha. Do you have a definition? So Doc Doe asked, do you have a definition of a stable starter?
Ooh. So again, I think I would personally describe a stable starter the best that I can at this point as a starter that behaves about the same way day after day or bake after bake. If, like me, you only bake with a starter every 2 weeks or so. So I'm defining stable based on the activity that I see of that starter or the bread that starter produces. If we are able to move forward and study these starters over time, then and actually look at the changes or the stability of the microbial membership over time, then I would define a stable starter as one that has the same bacteria and yeast present over time, also in the same kind of abundance. So two different definitions of stable, one that we can describe now and one that we would need a lot more data able to define.
Yeah. Let's see. Ross K asks, can you talk about the hooch on top of the starter? Does it have different things in it than the more solid elements of the starter proper?
Yeah, so as far as I know, a lot of that hooch layer is alcohol. So that's kind of when you get a really hungry or hangry starter. I see this especially in the fridge, like when I do go 2 weeks or sometimes between my bakes.
Shame on you.
I know there was a while I was not a very nice starter mother. I got really deep layers of hooch on my starters, and they do smell a lot like alcohol and acetone. Part of that is from the solid particulates, you know, like all of the flour bits are settling out over time, but I think a lot of that hooch layer is alcohol. I wouldn't recommend drinking it. It's not that kind of hooch. But yeah, it is, I think, referred to as hooch because it has all of that alcohol in it.
Reading these comments is awesome, guys.
Yeah.
It's great to see you guys answering each other's questions. Gosh, Erin, only every 2 weeks you use sourdough? I may bake every 2 to 3 days right now, or pancakes, waffles, rolls. Invite us over.
Well, so every 2 weeks I bake bread, but then I do make pancakes and waffles for sure.
I think that's the end of our questions. Is that possible? It's also the end of our time. Wow, that worked out really perfectly. Thanks for everyone for tuning in and for being a part of this project and for asking so many amazing questions that make us ask questions and think about it in a new way.
Yeah, this has been incredible. This YouTube video, we're going to try and edit out some of the stumbles at the beginning, and certainly that mute part that you helped us to catch. But aside from that, this video is going to be posted online like in perpetuity. Yeah, it's here online now, and it'll stay here for a while. So you can feel free to refer back and share it around. And we'll still be kind of haunting the Sourdough Project group on Facebook and sending updates as they come in. So,
and if you're sending in questions throughout the year to our Your Wildlife email, I'm sorry if I don't answer it quite as fast as I'd like to. It's mostly because I want to give your questions the time they deserve, which we're a small team of 2 here. So it takes us a little while sometimes, but we're always, even sometimes when I don't answer the question, I'll run over and be like, oh my gosh, did you see this person's email? So please keep sending us emails. We appreciate them even when we don't have time to answer them.
Yeah. Thank you for your time, for your starters, for your patience, and for your input and participation.
And all of your knowledge. I'm learning a lot. Thank you.
Yeah, it's incredible. So until next time, bye.
Bye.